Hello, everyone, and welcome to today's webinar, Uncovering Novel Immune Landscapes in Ovarian Cancer with Spatial Proteomics. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Akoya Biosciences. To learn more about our sponsor, please visit akoyabio.com. Now, before we begin, I would like to remind everyone that today's event is interactive. And we do encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you have any technical issues here, you can submit them in the Q&A box as well, and we will help you directly. Now, I would like to welcome our speakers, Riley Arseneau and Sarah Nersesian, both PhD candidates at Dalhousie University. For complete biographies on our speakers, please visit the presenter tab at the left of your screen. Riley, Sarah, welcome. So great to have you today. You may now begin your presentation.
Hi, thank you so much for that introduction, and I'm very excited to get started with our presentation today. We're going to first start off with some acknowledgements. Our university, Dalhousie University, and our laboratory, the Boudreau lab, are both situated on Mi'kmaq territory, which is the traditional and unceded territory of the Mi'kmaq people. And I also want to start off by recognizing the co-authors of this work and the associated publication that we'll be talking about today, which include a number of different individuals across Canada and a representative from Akoya Biosciences of Vic, who really helped us with allowing this publication to come to fruition. Without their very crucial contributions, we would not have been able to conduct this work. So today we're going to be talking about high-grade serous ovarian carcinoma, or HGSC, which is how we'll be referring to it. It's the most common and the deadliest gynecological malignancy. In part, this is due to its very high level of heterogeneity within and between tumors of the same patient. It also has resistant clonal outgrowth in response to the therapies we have available. It frequently is also able to evade adaptive immunity through the loss of HLA expression and targetable antigens. And all of these features also exist within a very immune suppressive environment. So today's presentation is going to talk a lot about immunology. I want to give a brief overview for those who may not be as familiar. So we'll start off with innate or natural immunity, which is untrained, meaning it's functional and effective without the need for any prior exposure. This category often includes innate cells, like our favorite immune cell, NK or natural killer cells. These cells can perform a multitude of functions, including direct cytotoxicity, antibody-dependent cellular cytotoxicity, and can also immunomodulate their environment through the release of immune signaling cytokines. Another innate immune cell, called a macrophage, is also important for immunomodulation. These cells can phagocytose or eat target cells and process bits to present antigens to train other arms of the immune system, which is also termed the adaptive immune system. Within the adaptive arm of the immune system, this includes T cells, which are important for cytotoxicity but require antigen recognition to perform that function. They're also important for immunomodulation, playing a role both through the release of cytokines that can activate other immune cells as well as suppress them. And it also includes B cells. These recognize antigens and upon recognition can produce antibodies, which when bound can signal to other immune cells that the target requires killing. Each of these cells has been previously reported to be prognostic within HGSC tumors. More infiltration typically predicts better outcomes and survival. However, these associations have been made independently. An important concept that we'll be talking a lot about today is the importance of immunity being a team sport. So we'll walk through this example and this process here. NK cells are important for direct cytotoxicity, as we talked about, and so they can provide killing to the tumor HGSC cells without any previous training. Macrophages within this environment, as I said, are important for phagocytosis and processing those HGSC antigens. Once processed, they can present antigens on the receptors to help train other immune cells, teaching them what to target. For example, B cells with the right receptor can then bind and become activated. These will then go on to produce antibodies against that specific antigen target. T cells are also trained in this way, using antigen and co-stimulation provided by cells like macrophages to become fully functional killers. Once these T cells are trained, they can then use those antigens presented on a target cell's HLA to identify that target towards killing. T cells therefore require HLA and antigen. This allows them to identify whether a healthy cell is presenting that antigen or a toxic cell. And the last feature that we'll be talking a little bit more specifically about today is antibody-dependent cellular cytotoxicity, or ADCC, which is primarily attributed to NK cells. And it uses an Fc gamma receptor called CD16A to bind to antibodies that are attached to a target. Signaling through that CD16A can then activate NK cells for target cell killing. Now we're an NK cell lab, so I'm going to talk a little bit more about NK cells. They're a unique type of lymphocyte that can detect hallmarks of cell stress and DNA damage rather than identifying specific antigens. So in a healthy situation, like on the left, these NK cells would inspect a cell, see it expressing MHC or HLA, which is an inhibitory signal that tells the NK cell that it's all good. When the cell starts to become unstable because of examples like transformation into cancer or even a viral infection, these cells will often downregulate inhibitory MHC or HLA, and this missing signal then poises an NK cell to look for more. For some NK cells, just losing HLA can be enough to activate them and kill their target cell, but most NK cells require a little more confirmation. So when a cell becomes really stressed, for example, a tumor cell, in the example of HGSC, they'll start upregulating NK cell activating ligands. And these will directly tell the NK cell that this cell is not okay and needs to be eliminated, which adds another important signal. So flipping back to this figure, we talked about the barriers to HGSC. These features and barriers are actually ways that NK cells can meet those barriers head on. Since NK cells detect hallmarks of cell stress rather than specific antigens, they can evade heterogeneity and can deal with resistant clonal outgrowth. NK cells can also facilitate antigen-specific responses through ADCC or antibody responses, but they can also use that HLA loss as an activating signal. One limitation of all immune cells within the immune microenvironment is how suppressive this environment is. These immune cells then become dysregulated and no longer functional. So figuring out how to support these immune cells within the tumor is an important task that still requires additional research. NK cells are very diverse. They're often and frequently split into two main categories: CD56 bright on the left, which are usually a more immature state that associate with cytokine production, while CD16A positive NK cells are more differentiated or mature. And these are the NK cells that are most often associated with cytotoxic functions. In reality, there are many different flavors of NK cells that exist between these two extremes. However, in this study, we were curious specifically to see how the CD16A mature and cytotoxic NK cell population impacts HGSC. This population of cytotoxicity is also capable of ADCC, which can be facilitated through antigen presented on an HLA, but it can also signal through directly binding antigens on target cells. Importantly, CD16A signaling is uniquely potent in an NK cell. It can stimulate NK cell killing even in the absence of other activating signals. This recognition of antibodies has already proven to have real clinical implications. For example, we know that different polymorphisms or small differences between individuals' Fc gamma receptors can influence treatment outcomes to some of the biggest antibody therapies that are available in oncology to date, including cetuximab, which is an anti-EGFR antibody, or trastuzumab, which is an anti-HER2 antibody. While our overarching goal in this project was to figure out how to better support anti-tumor immunity by figuring out which components were important, we were also specifically interested if learning more CD16A positive NK cells within the HGSC tumor before treatment led to better responses and outcomes, and also to find out which other immune cells were often found collaborating with these NK cells. So with these questions, we turned to multiplex immunofluorescence, and we developed a panel which could simultaneously identify multiple markers within the same HGSC tissue. So you can see these markers here. And our goal with this panel was to identify and resolve the two different NK cell subsets. For example, one that was expressing CD16A and one that was just expressing another canonical marker, CD94. CD94 was also expressed on T cells, so we added CD3 to differentiate those T cells from NK cells. We also were looking at CD16A, which is often also expressed by macrophages. And so to differentiate that myeloid population, we added a CD68 antibody, resolving two populations of macrophages. And to separate the regions of epithelium from the stroma, we also used a pan-cytokeratin marker. So we then used this multiplex panel to stain a subset of the Canadian Ovarian Experimental Unified Resource, or CORE cohort, which following tissue processing ended up representing duplicate tumor cores from 939 HGSC patients. These patients ranged in age from 26 to 91 years old with a median age of 62 years old. They were diagnosed between 1992 and 2014. This was just before PARP inhibitors were approved for treatment, so importantly, none of these patients actually received that treatment. We also saw that over 76% of these tumors were grade 3, representing what we see with HGSC tumors. And half of these tumors had residual disease following surgery. Over 80% were late-stage tumors, representing those that had already disseminated throughout the peritoneum. And at the time, only a few hundred were tested. Approximately 17% of those that were tested had known BRCA or BRCA mutations. We started by staining our HGSC tissues using the opal multiplex system and imaged them using the PhenoImager multispectral camera. Next, we conducted spectral unmixing in Inform, which allowed us to pull the signal from each of the markers within the tissues. Following that, we performed tissue segmentation. This allows us to separate the regions of the tumor into the epithelial compartment, which you see here in red, the stromal compartments in green, and any of the regions that were off-core in blue. Next, we used DAPI nuclear staining to segment and separate each nucleus, which then represents a single cell. These cells are then given unique cell IDs, with each cell ID associating with a unique pattern of marker expression. These patterns were then run through unsupervised clustering, which took into account both the physical characteristics as well as the fluorescent signal of each cell within the image. In this way, we were able to assign a phenotype to each of the cells within that tissue. Finally, we conducted spatial analysis, where we looked both at nearest neighbor as well as cellular neighborhood k-means-based clustering to see what cells were most frequently co-localizing with others. Our results were surprising to us in a few ways. First, we found a subset of T cells that express CD16A, and searched the literature, we actually found that a few papers had described this subset before in different pathological situations, including viral infections. Perhaps, though, the biggest surprise we observed was that upon unsupervised clustering, we resolved a single positive CD16A population that didn't express common lineage markers that would align with conventional NK cells, T cells, or macrophages. We had several theories as to what these cells might be, from some representing CD94 negative NK cells to others that had described CD16A positive fibroblasts or recently infiltrated monocytes. But we decided to move forward with our analysis to see what impact these mystery cells had and what those defined cells had in store. And now I'm going to pass it off to Riley, who's going to be talking about some of our early results.
Thanks, Sarah. I'm going to take over the results now. So first, we looked at the cell densities for each of the phenotyped cells by averaging the cell densities in cells per millimeter squared in each tumor compartment. So the S represents the stroma, while the E represents the epithelium. We also looked at the percent of patient tumor samples contained in these populations. So the majority of the tumor was infiltrated by HGSC tumor epithelial cells or non-epithelial, non-immune marked stromal cells, which likely captures other immune populations we just didn't specifically stain for. What we were surprised to see was that almost all HGSC tumors were infiltrated by our mystery single positive CD16A positive cells at the macrophage level densities. We also find lymphocytes to represent a smaller proportion of the immune compartment, which others have also described. So NK cells represented the smallest proportion, with CD16A positive NK cells expectedly being the rarest. Next, we compared the densities of each of the phenotype cell populations against the characteristics of the HGSC tumors they resided in. So when we looked at how these densities differed, we found that a higher density of the single positive CD16A population within the stroma associated with grade 3 tumors, and then stage 4 tumors versus stage 3 tumors. We were also excited to see that the stromal CD16A positive cells were enriched in tumors that responded better to treatment, more likely being chemosensitive following 6 months of therapy. We then conducted univariate Cox proportional hazard models and visualized them in the forest plots shown on screen. So to orient you, the left of the midline indicates a survival benefit, and to the right of the midline indicates an increased risk of death. Each box represents the hazard ratio, and the range indicates the confidence intervals. So we wanted to ask how these cell densities associate with survival, and we found surprising results. Most strikingly, in each immune cell population, there was a more dramatic survival benefit in the CD16A positive compartment. Having this Fc receptor seems to be providing a survival advantage, and even though they are not significantly significant, it would suggest that these populations independently may influence a response. We also noticed that having epithelial cells within the stromal compartments, likely representing early metastases, demonstrated worse overall survival. So now we understand that these CD16A populations were prognostically independent. But as we mentioned in the introduction, anti-tumor immunity is a team sport. So we wanted to ask how these immune cells are interacting together. The first question we had is who is often found infiltrating along these CD16A positive populations? So we used Pearson's correlations to assess co-infiltration. And here we're looking at how overall cell densities correlate within the same tumor. So we found that immune cells expressing CD16A positive co-infiltrated with each other, but rarely with the CD16A negative immune cell populations. We also quantified the nearest neighbors for cells from each population and observed that CD16A positive cells spatially co-localize within HGSC cores. So along the x-axis is each cell phenotype, and then along the y-axis is the proportion of the nearest neighbor phenotype. And to the right, we have the CD16A populations, and as you move from left to right, you can appreciate that the proportion of CD16A increases. So we next conducted a spatial analysis to cluster cell subsets into multicellular neighborhoods. So in brief, for each cell within each core, using the phenotype maps, the 15 nearest neighbors are counted and recorded. Then using unsupervised clustering, these patterns of co-localization were clustered into 6 unique cellular neighborhoods that varied in their frequency, distribution, and cellular composition between patients. So here we see 6 cellular neighborhoods, and 2 of these neighborhoods were devoid of immune infiltration, and predominantly they're epithelial or stromal. So we refer to them as the cold epithelium or the cold stroma, respectively. The remaining 4 neighborhoods were immune infiltrated. Among them, 2 were highly infiltrated with CD16A positive cells. So we have CD16A enriched stroma and then CD16A enriched epithelium. And the remaining neighborhoods were defined based on the extent of immune infiltration as immune-enriched or immune-moderate. So in these figures, we're showing the neighborhoods that individual cells within HGSC cores are associated with. So in the region in the first image there, you can see the cold stroma in the light purple, and then the cold epithelium in the darker purple. And we notice the patterns of spatial distribution between cores where CD16A neighborhoods were often enriched at the epithelial to stromal interface. Next, we wanted to compare cellular neighborhoods coverage against tumor characteristics. So as we can see, grade 3 tumors were occupied by more CD16A enriched stroma and significantly less cold epithelium, as shown in the first graph here. We also found that CD16A enriched stroma was the only neighborhood to differ between the stage 3 and stage 4 tumors, with stage 4 having a higher proportion of CD16A neighborhoods. Looking at chemosensitivity, we found that when occupied by a higher proportion of cold epithelium, the tumors were associated with chemoresistance. We then next looked at the proportion of neighborhoods against survival. So despite the fact that CD16A enriched neighborhoods were proportionally higher in the higher stage and higher grade tumors, they maintained a survival benefit as shown in the forest plots to the right. So we observed no survival benefit for the immune-rich stroma and immune-moderate stroma neighborhoods, indicating that this benefit seems to be associated with the CD16A positive neighborhoods and was not due to general immune presence. Which brings us to our next question, and I'll hand that back off to Sarah.
Thanks, Riley. So I am back to talk about some of our other questions. Understanding that some of those cells and neighborhoods influence response to chemotherapy, we were then curious to identify how chemotherapy could influence our identified cellular neighborhoods. So we had an additional small cohort, which represented 53 HGSC patients who had received chemotherapy prior to their surgical resection and biopsy collection. So we stained these samples with the same multiplex panel. Even within this small cohort, we saw dramatic differences in neighborhood proportion covering HGSC tumors following chemotherapy. We found that the area of epithelial neighborhoods that were infiltrated as well as non-infiltrated was lower in post-chemotherapy patients, while stromal neighborhoods were enriched. We observed specifically a decrease in the area of CD16A enriched epithelial neighborhoods in patients that had received that chemotherapy. Reflecting this depletion of CD16A positive neighborhoods, upon survival analysis, we lost that prognostic benefit, but we maintained an even stronger benefit for those who had a higher proportion of CD16A enriched stroma neighborhoods within their tumor, indicating that this survival benefit was still true after chemotherapy treatment. So our initial panel wasn't the most conventional, and we had designed it so that we could look at NK cells. We found surprising results, and therefore we wanted to contextualize those cells and neighborhoods with more well-established immune infiltrating markers. So therefore we integrated CD8, which is typically affiliated with cytotoxic T cells that are prognostically beneficial, and also CD163, which can identify suppressive myeloid populations. Here is our expanded opal multiplex panel, which is harder to appreciate in a disorganized HGSC tumor. So here we've shown our optimization process where we stain this panel on a human tonsil sample that can really show the highly regional specificity to our immune cells of interest. So here you can see the added immune cells that we hope to identify, which included phenotyping T cells more specifically into a CD8 positive population, and macrophages more specifically into a CD163 positive population. We again correlated these to look at co-infiltration. So that means which HGSC tumors had multiple types of immune cells infiltrating. So within this expanded panel, we again observed that CD16A positive immune cells co-infiltrated in terms of density across their tumors. And we saw surprisingly that both CD8 and CD163 immune populations were found within these clusters. We also again looked at nearest neighborhood analysis. And here, once again, we saw that the cells positive for CD16A most often co-localized and were close nearest neighbors to other immune cells expressing that CD16A. This was a small cohort, but we again wanted to look at what neighborhoods we could resolve. So we conducted the same spatial analysis as earlier. In this analysis, we extended our panel to reveal 8 cellular neighborhoods. Many of these neighborhoods had actually reflected those from our previous analysis. Notably, neighborhood 6 or N6 represented our CD16A positive enriched epithelium. Within this neighborhood, we see a high density of both those CD8 cells, but also CD163 positive immune cells. We see here that N0 and N3 mirrored the cold epithelium, while N1 neighborhood mirrored the previously defined cold stroma. We also observed immune that were non-CD16A positive enriched neighborhoods, both in the stroma as N2 and the epithelium as N7. And this was similar again to our previous analysis of immune moderate and immune rich respectively. Again, these were proportionally different between HGSC patients and covered a different area and proportion of the tumor. We again wanted to test the prognostic impact of these expanded neighborhoods. Here, we found that the N6 neighborhood was strongly associated with overall survival. Even though this was a small cohort, we were able to find significant differences between those that had a higher proportion of neighborhood 6 compared to those with a low proportion of neighborhood 6. In conclusion, we found that CD16A positive cells infiltrate almost all HGSC tumors, and their density associated with features of the tumor, including their grade, their stage, their chemotherapy response, and survival of those patients. We also found that CD16A positive immune cells co-localized within neighborhoods that featured CD16A positive NK cells, T cells, and also macrophages. And these neighborhoods most often were found at the epithelial to stromal interface that associated with better survival. We also discovered that neoadjuvant chemotherapy had shifted those neighborhoods from CD16A positive stroma to colder stroma. And interestingly, to contextualize our results more specifically, we found that CD8 positive, cytotoxic, as well as CD163 immunosuppressive cells were also found frequently within those enriched neighborhoods. So revisiting this core concept, while we observed that individual cells were often not prognostic as standalone biomarkers, when they were actually integrated into cellular neighborhoods, those with higher densities of CD16A positive immune cells, which represented those cytotoxic CD16A positive NK cells, T cells, and macrophages, were most predictive of outcomes. This really emphasizes to us that it's likely representing the presence of multiple features of immunity, including the different components of innate and adaptive immunity collaborating together to have an impact on survival outcome, which we're not really able to appreciate as independent markers. However, spatial analysis as well as multiplex imaging allows us to appreciate these collaborations more directly. This brings us to our thank you slide, and of course, we have to thank our supervisor, Dr. Jeanette Boudreau, as well as all of our lab members, including Stacy and Jorge, who played a crucial role in data collection and analysis. We also wanted to thank our supervisory committee and collaborators who have helped us on this project, and all our funding sources that have provided us funding to actually conduct this research. And lastly, we also would like to greatly thank and appreciate the different patients that had donated their tissues to allow this work to become possible. And we'd be very happy to take any questions and answer any thoughts.
Sarah, Riley, thank you so much for your informative presentation. It is now time for our live Q&A portion of our webinar. Now to our audience, if you have any questions you'd like to submit, do so now. Just type them into that Ask a Question box and click Submit, and we will answer as many of your questions as we have time for. So Riley, Sarah, we have some great questions that have already come in, so I'm just going to dive in, starting with this one. CD94 and CD16 are unconventional markers for identifying NK cells. Why did you choose these markers?
I can take this one. Yeah, so that's a really good question. And unfortunately, more than 70% of HGSC tumors actually express CD56, which is the most conventional NK cell marker within their epithelium. So CD56 or NKM1 is actually a really poor marker for HGSC tumors where it's actually expressed by the epithelium itself. So to kind of get that spectrum of NK cell phenotypes, we decided to go on either end, one which was CD16A, that was the more mature NK cells, and CD94, which should be canonically expressed by all NK cells at all stages.
Thank you. Next question. How did you conclude that 15 cells within a neighborhood was the appropriate amount of neighbors to consider?
Sure. So we had run through a range of different neighboring cells. We had used 5, 10, 15, and 20. And from this, we had generated the heat maps for the cellular enrichment. And what we wanted to do was look at the point where the different neighborhoods were most distinct, but had the cleanest cellular neighborhood frequencies there. And we've also been doing this recently with another analysis where we're looking at a range of microns as well for a radius. So we've done 30 through 150 microns. And we're, again, using the same type of analysis where we're looking at how the distinct the neighborhoods are from each other while also being the cleanest there.
Very good, thank you. Next question. Do you think CD163 co-localized with CD8, meaning exhaustion of CD8 T cells?
Yeah, that's a really good question. I think that in the CD16A enriched neighborhoods where we also saw CD163 and CD8, it's hard to comment specifically on the phenotype of those CD8 T cells. Typically, we would think that if they're in these regions of high immune infiltration, that they're probably being stimulated and engaging with the tumor as well as other immune cells. So it wouldn't necessarily be wild to conclude that they're probably exhausted. But something interesting that we also noticed was that the CD163 cells, which are not usually prognostic, when they express CD16.8, and we didn't show this in our presentation, but it is in the last figure of the paper. So where you see the 163 cells also expressing CD16A, they're actually more prognostic, kind of leaning towards a survival benefit compared to those that are just expressing CD163. So that could suggest that those cells expressing CD16A have a unique type of feature that maybe allows them to be more immunogenic or more responsive to different kind of elements like antibodies.
Thank you. How did you validate the computational aspects of tissue and cell segmentation?
So we worked with a pathologist, a board-certified pathologist, who essentially we blinded to the different computational analyses. And what we did was we provided him with a range of, for example, the tissue segmentation from 100% epithelial cores to 100% stromal cores and kind of everything in between to provide him with kind of a variety of what the different tumors might look like. And what we asked him to do was actually outline the regions which he would classify traditionally as epithelium and separate that from the stroma and the off-core regions. We also got him to identify any blood vessels, so those highly vascularized regions. And what we did then was compare those regions that he had identified, and we looked at the concordance between what Inform had classified as those regions, and we found almost over 90% concordance when it came to tissue segmentation. And we also did the same thing when it came to cell segmentation, where we looked at the number of nuclei that were being counted within the regions. We found that it typically was really good at identifying epithelial regions with those larger nuclei. However, we also noticed that Inform had about a 10% over-enrichment of counting in the stromal region. So we had to take that into account within our analysis as well as our interpretation. But it was good to have that extra kind of validation from a pathologist to make sure that what we were calling single cells, what we were calling to tumor regions through Inform were actually what a pathologist would also assign to those regions.
Thank you. And Sarah, Riley, we have time for a few more questions, so I'm going to keep going. What approach did you use to conduct phenotyping in an unsupervised manner?
Yeah, so we had used FlowSum in our studio, which takes into account the expression profiles as well as morphological features of the cells, and then it uses this to cluster or unsupervised cluster into groups, which are the phenotypes it identified. So in the first panel, we had 40, and then the second panel, there was 43 different clusters. And then from there, we had gone in and reviewed the phenotyping that it performed and reclassified them into the phenotypes that we identified in our panel.
Thank you. Next question coming in. Hi, great presentation. I wanted to ask if in the future, will you be considering developing a co-culture organoid model to see interactions of CD16A positive NK cells and other immune cells?
That's a great question. And we actually have a peer in our lab, another graduate student, Morgan Prutul, who is actually developing something she calls T-Slice. Publication should be out quite soon. And in that model, we're able to create HGSC organoids and spheroids within different types of tumor environments, which can also include different kind of thicknesses of the extracellular matrix components. And we've already started working with co-cultures that look at interactions between NK cells and different myeloid populations within that kind of model system. So definitely something we're interested in doing.
Now, piggybacking on this question on CD16 positive, A positive, why do you think CD163 cells that are traditionally considered immunosuppressive were frequently found within CD16A positive neighborhoods?
That's another really great question. I think that what was kind of unappreciated previously was the diversity of myeloid cells that can actually exist within a tumor. And we're not necessarily the most myeloid lab since we focus on NK cells. But reading the literature, I definitely have learned a lot more that, while macrophages were traditionally classified as M1 and M2, there's a lot of different ways to classify them, including looking at their metabolic profiles or looking at some of the epigenetic changes that they experience. And I think that there's just more nuance than macrophage kind of dichotomization, and what these CD16A positive, 163 positive macrophages are is probably something distinct from what we would classically call and classify an M2 macrophage as.
Thank you. I do want to thank both of you, Sarah and Riley, for your time today and your important research. I also want to thank LabRoots and our sponsor, Akoya Biosciences, for underwriting today's educational webcast. Before we go, I do want to thank our audience for joining us today as well and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period, they will be addressed by our speakers via the contact information you provided at the time of registration. Pearls, I see one more question that came in that I think might be valuable to ask. Are you good to do one more?
Yep.
Okay, let's go with this one real quick. Now, did you say that you've been able to show statistically significant association between co-localization of the CD16 positive NK cells and some of the other immune cell types and survival therapeutic outcome?
Yeah, so that's another really great question, and that's exactly what we saw. So while we saw the NK cells that were only CD94 expressing, so those without CD16, we kind of saw them as loners within the tissues. They weren't frequently co-localizing with other immune populations, and they typically were also not co-infiltrating, so both within the same tumor and also within neighboring cellular compartments. And so all the associations that we saw with survival benefit for therapeutic response were those that were specifically the cytotoxic NK cell subset, which also happens to be the rarer subset within the tissue tumors within HGSC.
Great. Thanks for squeezing that one in. Now, I want to remind our audience as well that today's live webcast will be available for unlimited on-demand viewing following this presentation, and LabRoots will alert you via email when it's available, and we encourage you to share that email with your colleagues who may have missed today's live event. Now, Sarah Riley, before we go, do you have any final comments for our audience?
Thank you for coming and listening to us. And yeah, just check out the publication and email us if you have any questions. We're always happy to chat and collaborate.
Excellent. Very good. Thank you both again for your time today. We appreciate having you. And thank you everyone for joining us. We hope to see you all again soon and have a great day. Bye-bye, everyone.